Hi all, this is Mr. Labonte. I am going to be doing hopefully a quick presentation on genetics, starting with my obligatory cartoon about gene splicing. All right, there's a bunch of different terms to understand for genetics. The good news is you know a lot of these terms. So obviously you know a gene, sequence of DNA that codes for a protein. Um, it's important to understand the difference between a genotype and a phenotype. So a genotype is the genetic makeup because we are diploid. So let's just say we have R, say red eyes, little r, white eyes, or something like that. So we're diploid. One chromosome is going to carry that. The other is going to carry that. And the notation, the letters, are just whatever we want. The phenotype is what the organism looks like. So this organism may have red eyes, right? So that's the physical trait of the organism. The whole purpose of this is understanding about gametes. We just went over that. That's a haploid cell, right? And then the allele is one or more forms of a gene. And I'm going to hit this a lot, hopefully, during this and the next presentation. So right now, you probably have learned that you could have like, whatever, sorry, let's stick with our R's. You could have R, you could have R. It's also possible that you could have R, another allele. So that could be like red with spots, or you could have like R prime, right? So you don't just necessarily have two alleles like capital, lowercase. You could have a cursive, you could have a prime. So you could have multiple alleles for a particular gene, and we'll talk a little bit about that. Hopefully you've heard these terms before. Homozygous, same. So that would be like capital R, or lowercase r. Heterozygous, different, capital R, lowercase r. And then dominant, which we tend to describe as the capital. So if one allele is expressed over the other, recessive would be the lowercase one that's not. And we may go over true breeding, wild type, incomplete dominance, polygenetic, and things like that, but I'm going to skip through those right now. All right, so Mendel was the person who really came up with the math and the process by which this happens. And what he was looking at are pea plants that were breeding with themselves. So if you have a true breeding organism, this white pea plant that breeds with itself would always have white babies. And so for what, just because the R's are easy, we'll say that is recessive, lowercase r, and the purple would be dominant, capital R, capital R. Hopefully you will have remembered doing these in maybe middle school. In this case, what you're looking at is what's the probability that you're going to end up with an offspring with a particular trait. Well, in the first generation, if this is classic inheritance or classic genetics, the dominant covers up the recessive. So you might have, I'm going to zoom in here a little bit. You would have this, say that this is the male. It could give a capital R sperm or a capital R sperm. This is the female, a lower R egg. And the sperm and the egg are going to meet. And the offspring, so these are haploid individuals, right? These are haploid cells, haploid eggs, and haploid sperms. But the offspring will be diploid. And that diploid will be all heterozygous, so capital R, lowercase r. So the genotype is the same and the phenotype is the same. And in this case, that's 100% purple, if we're going to use the example that we just did. However, if this organism here had a baby, then when it underwent meiosis, the cells could either be capital R or lowercase r, and the egg cells, so the sperm cells could be that, and the egg cells could be capital R, lowercase r. And that's going to give you a new diploid organism that could have 3 different genotypes, right, which would look like that, but only 2 different phenotypes. So the physical traits would be either purple. These would also be purple if it's classic genetics, right? And then that one would be white. So that's 75-25 phenotypic ratio. And these are the things that Mendel found. And the reason we call it classic genetics is because This is where he was at. So Mendel was identifying all of this. He knew that there was inheritance, and so this is a law of inheritance that he was trying to discuss. All right. And then what I just did was the Punnett square, which was created by Reginald Punnett. And so that was a mechanism of doing that. So I'm going to see if I can have you guys do one here. Let's just do one. Let's say, let's use the letter B. So this is individual who is heterozygous for bald. So let's say they're not bald. So capital B, not bald. And they mate with an individual that is homozygous recessive for bald. So this individual would be bald. So what would the offspring look like? Take a look at that and then hit pause, try that, and then I'll do it for you. All right, so let's take a look at this. Let's just assume it's the male. The male by convention is on top, probably because it's a sexist world. Female, so these are the haploid cells that are meeting. Sperm and egg will meet. 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 50% will be bald. 50% will be hair. And, could be a person or a fly. It doesn't really matter. So this is Punnett square, and this is how you figure this out. So this is for one trait, right? And when you have a baby, we just talked about having over, I think it was 8 billion different possible combinations. You have millions and millions and millions of traits. So we may want to be a little more complex. Actually, I'm going to skip recombination frequencies. We don't really have time, unfortunately. Because just briefly, I'll just say multiple traits are going to be on this same chromosome, and their distance away from each other is going to depend on whether they travel together. But here we are. Now you might want to have more than one trait. So this is G and Y. It doesn't really matter what the traits are. So this individual, this is an individual who is heterozygous. So the dad and the mom are both heterozygous for the trait. Now, the reason I like this slide is it shows, if you remember, this is independent assortment. The reason you have to do Punnett squares is you don't know what chromosomes are going to line up. So you may end up with an individual that looks like this. You may end up with an individual that looks like this from the same genotype. So you have to represent that. Here's a possible, these are the sperms that are possible from this organism. So you could have GY, right? GY, GY, so, right, that would be this one, or this one going up there, and this one, or GY. So these are all of the possible combinations that you could have when you have two organisms, one organism that undergoes meiosis. So when this organism undergoes meiosis, any of these could be possible. So now if you have a mating of two different organisms, right, the same thing, let's just assume for ease that this is the same genotype. Sorry, that's a G, capital Y, and lowercase g, lowercase y, okay? And then when they meet, right, this individual is going to get genes from both parents. I'm just going to do a few of these. That's a Y, 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 little g, big Y, and little g, little y. And there are 16 genotype squares here, but the options really end up being, for genotypes, capital G, capital Y, capital G, lowercase g, capital Y, lowercase g, capital Y, capital G, lowercase g, capital Y, lowercase y, capital G, lowercase g, lowercase y, and then lowercase g, lowercase y, lowercase y. So these are all the genotypes, and those combine to a phenotype ratio of 9, 3, 3, 1. We'll talk about that a little in class. So these are the phenotypes here. And I'll go over the example in class of what that would look like. You can see it here. Sorry, I forgot I included this slide. So this is with wrinkly seeds and round seeds. This is exactly what I just went over with the G and Y, except now it's R for color. So green and yellow, and then smooth and wrinkly are the two traits that are being presented here. So you can look at that and you see that these are the phenotypic ratios down here. All right, when you get to more than two traits, it becomes too difficult to try to make squares like that. So what you do is you could just do separate dihybrid crosses and then multiply them. So here you see you have AA, so that would be AA. So sorry about that, AA for a single monohybrid cross. And then you can write out what are the possible combinations. So you see you have 50-50, and that's exactly what's represented here. Then you do the same thing with the BB, and then you do the same thing with the CC. So you get all of the different combinations here. And then I'm going to erase this and clean it up a little. Then when you want to look at what a potential outcome is, so the question might be, what is the likelihood of getting capital A, lowercase a, capital B, capital C, capital C? You just trace it along. 1, 2, 3, and you multiply it. Capital C, it's this one up here. 1/2 times 1/2 times 1/4, that gives you 1/16. So when it's complex, that's how you do it. All right, a couple things that are important for you to think about. These are errors that are often made. So dominant does not necessarily mean more frequent. So the example I often use in class is hexaploidy. I guess I could use H versus H. So hexaploidy is having 6 fingers. Being 6-fingered is dominant to being 5-fingered. But clearly the vast majority of people on this planet are 5-fingered. It's a very rare mutation to have hexaploidy. But what it does mean is if you do have the hexaploidy gene, you will have 6 fingers. That's a certainty. So it is not necessarily more frequent, but when it's there, it shows up in the phenotype. The other thing, and we'll go over this in class, is dominant traits don't, actually I'll go over it a little bit more later, they don't take over and become more common if all things being equal, if there's no advantage. The frequency of the alleles stays the same. And that should be in the next slide. Yeah. So the example of this, like showing this, is this concept called the Hardy-Weinberg principle. Okay, and this is hexaploidy right here. So as I said, in 20 generations, the frequency of hexaploidy is going to be exactly the same. And the way that this works, assuming a few different things, there are some assumptions about this, but what it shows is that the frequency of alleles is always going to stay the same. So what you can think about is you can think about Q being one allele, usually this is recessive, and P being another allele, and this is the dominant version of this allele. And so hopefully what you can see here is with P, you would have, like, let's say this is hexaploidy we're talking about. Oh, sorry, let me erase this stuff. I'm going to move this over so I can actually write on this. So if it's P squared would be H, Q squared would be little h, and then the heterozygous would be here, so the 2PQ would be 2 times capital H, lowercase h. So this is the math that shows for this, basically for 2 alleles, you're going to have a probability of basically it being 1 if you take each of these, homozygous dominant, heterozygous, and homozygous recessive, all of those together. And this, this image here, I can't turn this for some reason, this image here shows the probability of having each of these as the frequency goes up. And I will talk more about this in class. All right, just a couple more things. All right, another thing is that sometimes you don't have complete dominance. So you have the intermediate phenotype that shows. So this is what Mendel saw, and in this case, if you had, let's say, RR, that was purple. So this individual here is purple. However, there are many, many other organisms where the intermediate phenotype is its own, the intermediate genotype, or the heterozygous genotype is its own phenotype. So in this case, you have white and red combining, and when you have heterozygous, you have pink. And this is called incomplete dominance. That is not the only option, though. It's possible that rather than having the white and the pink gene mixed together, this white and the red gene mixed together to make pink, they could both be displayed. So here there's white and here there's pink. So again, this would be like an RR individual, and they would both be displayed. We have this when we look at blood types. So we can have A blood type, I circled the B. A blood type, B blood type, and AB blood type. So this is incomplete dominance. And we can have neither A nor B show. This has to do with antibodies, so you probably are somewhat familiar with this. And we've gone over this in class. So if you have type A blood, what happens is you make an antigen that has a particular shape. And in that self-testing that happens in the thymus, remember I said you could make any antibody ever, and then your body tests it against yourself. All of the cells that make A antibodies are destroyed, so you would have no A antibodies in your system, because it would be like, oh, I don't want to make that antibody because I'll kill myself. But if you don't have type B blood, you will make type B antibodies. Similarly, if you have type B blood, you'll make type A antibodies. Finally, if you have type AB blood, you'll make no antibodies, neither A nor B. But if you have type O blood, that is, you have none of these surface proteins, you will make both antibodies. And that's why you can only get the specific types of blood from each of the donors here. So that's an example of that. Finally, there are some traits that are carried by the chromosomes, by the sex chromosomes. So I hope that you are familiar with the idea that it is the father that donates the the Y chromosome, and the mother who donates the X. So whether you have a daughter or you have a son, the X chromosome is always going to be donated by the mother. So if there is some type of a mutation or a unique trait that's on the X chromosome and you have a boy, that boy always will have gotten that trait from the mother. Similarly, if there's some kind of mutation on the Y chromosome, which is basically things like testicular cancer, the boy will have always gotten that from the father, and the daughters will never receive that because they only get the information from the X chromosome. That has a lot of implications for why it is that females are generally more healthy than males. And then the very last thing is that sometimes you have multiple genes that contribute to one phenotype. So in this case, both A, B, and C are all contributing, in this case, to skin color, right? So the likelihood that an individual is going to be homozygous recessive for all three of these traits is very low. Similarly, the likelihood that this person is going to be homozygous dominant for all three of these traits is very, very low, 1/64th in both cases. But the chance that they'll have some variation, so they may have capital A, but they might have lowercase b, lowercase c, capital C, lowercase c, or they could have lowercase a, capital B, lowercase b, lowercase c. Hopefully you see that if these alleles are all contributing to the same trait, then these individuals are going to share the same phenotype. And this is polygenetic inheritance right there. All right, I know that was fast. I'll go over it in class, but I wanted to give you a primer on genetics.